There are a lot of different groups of invertebrates. They are the most abundant animals in terms of species, with well over 95% of animal species, and individuals, well over 99% of individuals. And certainly arthropods compose the majority of animal biomass. And so we certainly want to discuss invertebrates and their diverse groups in biology courses. It then makes sense to discuss the different groups in a typical order, so not at random, but is there some sort of order? The problem is that there was an order based on the relationships of invertebrate groups, and primarily those which are bilaterally symmetrical. So for example, this nematode worm, its head half is different from the tail half. And so you can't cut it in a transverse section and have two equal halves. The only way to cut a bilaterally symmetrical animal, a bilaterian, in two equal halves is to make left and right halves. That's what bilateral symmetry means. So we want not only does our nematode worms bilaterally symmetrical, but so are all of the vertebrates. How does one break these into groups? Well, the original classification, which was based more on anatomical structures, stated that the most primitive animals were like those of sponges, which don't even have tissues. Cnidarians have tissues but not organs, and they're radially symmetrical. They don't have a brain. Then there were primitive bilaterians, so here's a hydranidarian. Primitive bilaterians like flatworms, they don't have segments, they don't have blood vessels, they don't have a complete digestive tract, so that their mouth is still in the center of their bodies, and the same opening serves as both the mouth and the anus. And then there would be bilaterians which were considered a little more complex, like nematodes, which don't have segmentation, nor do they have blood vessels. Then other groups of invertebrates, so for example, nematode worms, have blood vessels. And then you would have the highest groups based on those which had blood vessels and hearts, which had a body cavity known as a coelom. And then we would split the coelomates into the protostomes and the deuterostomes, depending on whether the first embryonic pore developed into the mouth or the anus and other features. And so this was the original classification. As more and more genetic sequences were provided, however, this was revised. And what I'm about to show you aligns with the genetic data. However, it then makes a little less sense when it comes terms to the anatomical data. And so for at least, when it comes to teaching, it makes sense to talk about these flatworms, early, because they seem to be so primitive. Now, while there are still a few flatworms known as the acial or xenocoelomorph flatworms, which do seem to be basal bilaterians, the genetic sequences now include the flatworms, which were once thought to be very primitive, in with things like mollusks and annelids. And then nematodes get associated with arthropods. So there's a new group called the Lophophorates, and a second group, the ecdysozoa, because of a shared feature of molting. So the genetic sequence data aligns with this. But that gets us into problems, once again, with the anatomical features, which not only is important for biologists in general, but for a general biology student who's more likely to be looking at the anatomy than the genetic sequences. There are some problems. So for example, a previous video talked about flatworms, and the next videos we'll talk about mollusks and arthropods. And today I want to talk about all of the protostome bilaterians other than the flatworms, the mollusks, and the arthropods. So today I'm going to talk about diverse grouping. And one of the topics which comes up early is that of segmentation. If you want to build a body, an easy way to do it is if once the genetic mechanisms for developing a segment of a body is developed, that you simply then repeat this. So here the snake, much of its body then is repeating elements. When we look at embryos forming, here you can see are these blocks of tissue known as somites. So vertebrate embryos, even if we don't look as segmented, we develop these blocks of tissue known as somites, which repeat, and these can then develop into ribs and muscles and blood vessels, etc. The next group of invertebrates I talk about in this video will be the segmented worms, the annelids. And so you can see that not only are snakes and other vertebrates segmented, but that some invertebrates are segmented. Clearly this millipede is segmented. So here is a chicken embryo. And once again, you see those blocks of tissue known as somites. Here's a pig embryo, and you can see the segmentation there. So as we start talking about, and then even though it's kind of hard to see sometimes because the segmentation can be modified, as in most mollusks, when you look at human muscles, such as our rectus abdominis, it still comes in segments even though it doesn't have to. Mollusks have modified their segmentation in most of them, although there are still segmented mollusks. And so segmentation is a feature. So here's an early mollusk, which was looking like a segmented worm, which had a segmented shell. Segmentation is widespread among not only animals, including the vertebrates, but then also when we look at the invertebrates, because there are segmented worms like earthworms and leeches. There are segmented arthropods. So here you can see the segmentation in a crayfish. Crayfish then have segments in their limbs as well. And as I said, not only do fossil mollusks show some segmentation, but there are mollusks alive today known as chitons, which have segmentation or can have a segmented shell. And so even a lot of the organisms which we don't think of as being segmented, whether it be mollusks or humans, have varied this. There is segmentation in the arthropods, the annelids, the basal mollusks, and the vertebrates, etc. And so in the old system, the most reasonable explanation for why these animals typically have segments and these animals don't, flatworms aren't segmented, nematodes aren't segmented, is that segmentation evolved in the common ancestor of the animals that had a coelom. That makes sense. It's what's called parsimonious. There's a principle of parsimony, where the simplest explanation is often favored. But now in this revised classification, the genetic evidence supports this. But then here's the question. When did segmentation evolve? Because now these are segmented, the first ones of these are segmented, these are segmented, these are segmented, but those aren't, the brachiopods aren't, the nematodes aren't, flatworms aren't. And so did segmentation evolve multiple times? That's not as parsimonious. And it doesn't seem as if it evolved multiple times, because if you ask what genes does a vertebrate embryo use to establish segments, some of them are homologs of the genes that some of these invertebrates, like arthropods, say fruit flies, use to establish segments. And so if segmentation arose separately, you wouldn't expect multiple lineages to establish segments using the same mechanism. Well, so then if it didn't evolve separately, that means that it would have evolved down here, all right, genes for segmentation, at least precursors to segments, but that flatworms lost them and nematodes lost, my fault, and nematine worms lost them, and nematodes, I'm sorry, they're over here, lost them. And so this is a more complex family tree. And so once again, a difficulty in talking about some of these classification is that we can establish classification groups based on anatomical evidence, based on genetic evidence. But if we're using different types of evidence, what happens if they don't 100% agree? Well, then you ask more questions and you do more studies. But now I'd like to start with the segmented worms, the annelids. And I'm going to go through once again just kind of a set of the bilaterian animal groups, and then do mollusks and arthropods in another in other videos. So the bilaterians, they invented a brain, right? So sponges, cnidarians, they lack a brain. Now these brains are small, but nevertheless, if you're always going forward, then all of your senses are in the head area. It makes sense then to have a condensation and a collection of nerve cells. Now, interestingly, and I'll talk about this in a subsequent video, most of what we call the protostomes, they are the animals that have coeloms in which the mouth develops from the first embryonic pore. They have a nerve cord along their ventral side, their belly side, and it's solid. And this will be different from the vertebrates, where the nerve cord is along the dorsal side and it's hollow. And so worms do have a nervous system, and we can see some segmentation in it because each segment then has a bulge where you can find the soma of neurons, known as a ganglion. So this is a segmented earthworm, and the brain would be in that region. Earthworms have a complete digestive tract, unlike the flatworms of the previous video, in that food comes in a mouth, it passes through a pharynx, and muscle contractions in the pharynx are important in drawing food into the mouth. There is an esophagus region where the pH can be balanced and ions can be balanced to facilitate digestion later. And then the food passes into these two regions, a crop and a gizzard, where the food can be ground and particles crushed. So you can have digestive enzymes to digest the foodstuff, but if it's in the middle of this big particle, your enzymes can't reach it. So you need to grind up larger particles so you could access, say the organic molecules of, rotting leaves, for example. And then from here, the food passes into the intestine. And then it will then stretch the length of the body. And in the protostomes, the intestines end where the body ends, so that the anus is at the very tip of the body. That'll be unlike the vertebrates. But note the difference here between this group and flatworms, in that there is a complete digestive tract. Food comes in one end and out another, as opposed to, say, jellyfish and flatworms, where food comes in a centrally located mouth, gets digested, and waste leaves through that same opening. So that was something that bilaterians developed, which then would be inherited throughout the various branches of bilaterian groups. Now, flatworms lack a circulatory system, and that's one of the advantages of being flat, that, for example, diffusion, with the water surrounding you, could reach many cells if you have a flat body shape. However, if you're going to be larger or thicker, diffusion alone cannot distribute materials the way that you need. And so that it was the bilaterians which invented blood vessels. And in this earthworm, we notice there's a dorsal blood vessel which brings blood towards the head. There's a ventral blood vessel which pushes blood away from the head towards the tail. And along the way are contractile blood vessels, which we call hearts. all the heart really is a contractile blood vessel, which then establishes pressure to push the blood um, within. Now, as I'll mention later, there are different types of circulatory systems. In some, the fluid stays within these vessels the entire time, and therefore it's called blood, and these are called blood vessels. In others, in what's called open circulatory systems, there comes a point where the fluid leaves blood vessels and then mixes with the fluid in the rest of the body, including the coelom, in which case the fluid isn't called blood as much as hemolymph. Now, the coelom is a body cavity I'll be mentioning, and there's fluid here, and earthworms, in addition to having blood vessels, this fluid can then help to distribute materials, because as the earthworm contracts and moves, this fluid then helps to disperse materials as well. So in addition to the circulation material through blood vessels, which earthworms have, there can also be the distribution of materials through the coelom. This is known as a coelomic circulation. Earthworms, once again, this would perhaps be the easiest of the segmented worms to study. They have obviously a number of systems. The reproductive system involves both individuals being hermaphroditic, having both testes, which can make sperm, and ovaries, which can make ova. However, just because one has the gonads of two different genders doesn't mean that one can fertilize one's self, although in some animals that can happen. So here you have two hermaphrodites which are going to mate and fertilize each other. So each of them has sperm being made in the testes, and because the sperm are going to be released with a lot of fluid, you see these very prominent seminal vesicles producing the fluids which will be transported with the sperm. The sperm are then passed into these ducts, which then exit the body and then will be stored in what are called seminal receptacles in the mating partner. And so many animals can store sperm. Many vertebrates can store sperm. I mean, so when one considers, say, reptiles, a female reptile might become pregnant long periods after mating, even years, because the sperm were stored in her body. And so sperm leave from the male genital pore and then enter these seminal receptacles. When the individual then wants to produce the eggs, then the ovaries can be ready to produce these eggs. And this region here, the clitellum, produces a mucus secretion. And as it passes over the body, it will pick up the ova released from the ovary, and then it will pick up the sperm from the sperm receptacle. And then inside this mucus, then the sperm can fertilize the ova, producing the new embryos, which will develop into new segmented worms. And so this is the reproduction of earthworms. Earthworms are annelids, segmented worms. So when we break the invertebrates into groups, they are segmented worms, annelids. They are called protostomes because like arthropods and nematodes, the mouth develops first in embryos. And in the revised classification, they are in the Lophotrochozoa group. Now, earthworms not only include, annelids not only include earthworms, which are very important as decomposers. It also includes leeches, which can be ectoparasites, or they can be predators. They don't have to be parasites. And then it includes a lot of additional segmented worms. And so if you were to do a study of a freshwater body near your house, you might find oligochaete worms, which have these chaetae, these bristles, and they feed on detritus, bacteria, algae, and these are very small worms, and these that are important in food chains, so that small arthropods, small fish could then feed on them. And so, once again, of interest if you're interested in invertebrate groups, but also they play roles in food chains. So there are a number of different segmented worms. Now, those oligochaetes are on the smaller side, and here you can see leeches. There are diverse leeches. Once again, some are parasites, so that they can attach to a host, typically a vertebrate host, and they then extract blood from the host. They often secrete an anticoagulant so that the blood doesn't clot. So if you've ever had a leech on you, and I've had plenty, then you take the leech off, but it'll bleed more profusely than a usual cut, because once again, the leech produce this anticoagulant. Now, here you can actually see a type of parental care in leeches. So those are all young leeches attached to a parent. And this parent is going to go out in search of a host, say a snapping turtle. And upon finding the host where the parent can feed, then the young can detach and then get established on a host as well. And so in transporting the young to a host, this is a source of parental care. Many vertebrates, like many fish, offer zero parental care. So it's interesting. And here we see some of this in vertebrates. Certainly, segmented worms are a very old group. So if one goes to the Cambrian period, one can find a variety of fossil worms which are classified as annelids, including some which had some degree of protection. One last thing on annelids. Obviously, vertebrates have limbs, and we can talk about the limbs of fish and amphibians. Arthropods have limbs, segmented limbs. The jointed limb is what the name arthropod literally means. And if we were to then ask, where do limbs come from? One thing that should be noted is that if you ask, what genes does a body use to, say, make a limb here? So obviously, an insect's leg and a vertebrate leg are completely different. So, fish fins and tetrapod limbs did not evolve from the legs that, say, an insect has. There's no connection there. Or virtually no connection, because if you look at, say, fruit flies, the genes that fruit flies use to, say, put a limb here, many of them are actually the same genes that, say, a vertebrate uses, like a fish, to, say, put a fin here. So how could that be possible? Well, an explanation seems to be that perhaps before there were limbs that we would say, that's a leg, that nevertheless there were specializations in lateral abdominal walls, which then required some genes. So here is a segmented worm. And notice what it has along its segments. So these are not legs, right? And some of these structures can be used for respiration. But some of them can actually help to push the animal along. And so, in addition to things which are full-fledged legs, there also are other structures which can then serially be produced along the length of a body. And it seems that some of the genetic programming for make a leg here was a modified version of the genetic programming that animals without legs then used to, say, put something here, which could develop into what are called parapodia that you just saw, or maybe ultimately then be advanced into a limb. So If the same genes like DLX and others are used in vertebrates, in arthropods, and in annelids, then that would suggest that the common ancestor of the bilaterians had these genes. But then the problem is we don't see those yet, as far as I know, they have not yet been described, in things like nematodes or flatworms. And so once again, those classification schemes, they then, some are more consistent with genetic evidence, others more consistent with anatomical evidence. Fruit flies and vertebrates share a few more genes, including the signaling molecule sonic hedgehog, which is a very important signaling molecule used in both. Segmented worms, or annelids, are a group of invertebrates called protostomes, and then and within the protostomes, we could put them in the group Lophotrochozoa. There are so many other invertebrate groups, many of which are minor members. Now, one group alive today, the Priapulid worms, they are a minor group of worms. They're segmented, they're predatory, so they dig in burrows and they can come from the burrow and trap their prey. But in the Cambrian period, starting 540 million years ago, they were a major group of predators. And so one could discuss different groups from different angles. They play roles today, some more important than others. They have fossil representation. And one of the difficulties then is if there is disagreement based on different studies on how they should be classified. How do we compare, the anatomy of this one to this one if we're not, quite sure of, how closely they are related. So Priapulid worms, that's another group of worms. While many of the early bilaterians were worm-like, there are some variations on this. So there is a group of animals called rotifers. Now, you need a microscope to see a rotifer, or perhaps they're just visible to the naked eye. But these are complex animals. They're more advanced than sponges, than cnidarians. They are more complex in many aspects than things like flatworms, even though the flatworms can be larger than them. And so rotifers have this ciliary structure, which then creates a current. And here you can see this rotifer, both circulating the fluid around it in the pond and pumping it, so that it can then filter feed and use the water as it passes to capture bacteria, cyanobacteria, algae, and other things on which it feeds. And so even though this is a complex animal, there are one-celled paramecia which are bigger than many of these rotifers. So that's interesting that you can have one-celled animals larger, one-celled eukaryotic protists larger than many animals. So notice they're bilaterally symmetrical. They have a complete digestive tract. They can, at least. We then consider that to be more derived in the situation you would say have in flatworms. The females may carry an embryo for a while. So once again, here's an invertebrate offering some degree of parental care. You can see the embryo of the rotifer there. And once again, some biologists might be interested in rotifers for their anatomical features, or how they relate to other organisms in grouping. Once again, there's that embryo. But another reason to be interested in them is once again just the role in food chains that they play. So they have a mouth, an eye spot. They possess chitin. So chitin is something which we can find and not only in fungi, but also in the arthropod exoskeleton. Here you see structures which are made of chitin, which help them process food. This foot region can help them attach to a site. And the corona then has the cilia, which then helps them feed. So here, once again, rotifers are common in aquatic environments where they play important roles for food chain. So once again, even if you were interested, say, in more advanced animals like fish, young fish, they have to eat smaller things, especially early in life. And so rotifers are a group of bilaterians, as are nematodes. Nematodes are one of the most abundant kinds of animals on Earth, even though most people don't know them. That is because the majority of them are microscopic. So when you walk, say, on soil, you're stepping on nematodes, lots of them. But you would need to, take the soil, add a drop of water, and look under a microscope before you could find them. They have a complete digestive tract. Once again, this is simpler than flatworms. Unlike flatworms, which are solid, notice the space here. It's not considered to be a true coelom. I'll be talking about the coelom presently. It's what's called a false coelom or a pseudocoelom. But nevertheless, once again, that's kind of considered more derived than a flatworm. So they do have muscle. Under the microscope, you can recognize a nematode compared to, say, an annelid, because nematodes, they both have muscle which run the length of their body, longitudinal muscle. But earthworms and other annelids, they also have a second layer known as circular muscle oriented differently around the body, which just gives them more versatility in the movements that they can create. So the longitudinal muscle pulls on the cuticle, and the fluid in this pseudocoelom, in this space, then offers a resistance which helps the movement. And there is no hard structure like bones or anything, but once again, one of the things that fluid can do inside the body is serve as a cavity of resistance upon which muscles can pull or push. So once again, with only the with only the longitudinal muscle, they have one layer of muscle rather than the two that are found in in earthworms. So once again, that would be the longitudinal muscle there. Here you see lots and lots of eggs, and one of the reasons that these are of concern is that many wild animals, so wild animals in our area, can have nematode parasites, foxes, bears, but so too can domestic animals, such as, say, pigs. And this is then a problem because if you eat undercooked meat, such as undercooked pork, you could actually then ingest nematode worms, which then become intestinal or parasites throughout your body. And this is certainly a large, a great problem throughout the world. Now, it's commonly observed that in, say, a general biology 2 class, a lot of students might say, I don't care about these invertebrates. I'm taking this course as a prerequisite to go and study medicine. And I certainly understand that. However, let's imagine, for example, that we were to take the 7 billion, actually there's almost 8 now, but say 7 billion people on the planet and split them so that each of these represents 1/7 of the world population. Now, many people, when it comes to sanitation, have use of indoor plumbing and toilets. Some use a structure like this, where say there would be a pit in the ground with a concrete slab and say a toilet that could maybe have an enclosure, or maybe just even something that then looked like this, that there was a slab over the ground. Let's say that all three of these are called improved sanitation. If you ask what percentage of the world's population has access to some type of improved sanitation, that would be about 4 and 7. All right, so more than half, but only somewhat, more than half. 2.5 billion people live without improved sanitation. So this is beyond what they then have access to for sanitation. And about 1 billion people would practice open defecation. Now, this then becomes important for many reasons, but one is that this helps to spread infections. And when we talk about things like diarrhea, in many nations, this is an inconvenience. When one talks about parasites, that certainly sounds odd. But diarrhea is a major cause of death throughout the world, as are nematode worms and infectious flatworms. So a variety of different kinds of nematodes, flukes, tapeworms. These are major sources of human illness and major sources of death, where there are just millions, and in some cases billions, of individuals who have these parasites living inside them. They are very common, and so, for example, some parasites could be spread by, say, walking in stagnant water. And if you're, say, planting rice, rice grows in stagnant water. So then larvae of worms could be burrowing into your skin. If flies visit a latrine, and then land on some food or a table, where you're about to eat, eggs of some of those tiny parasitic worms could now deposit it on your food. Now, this is a bit run down. It looked better when I lived there, but I was in the Peace Corps and I lived abroad for 2 years. And so I had a latrine that I use. And if you look, the well is nearby. And if that seems odd to you, well, it's because you're used to a more privileged type of life. You saw the percentages. A very large percentage of human population lives in situations more like these, where diseases are more likely to spread. I had a number of parasitic diseases when I lived abroad and had worms, and that's normal. And my point for saying this is, if a general biology student was thinking that I would like to study medicine and to help people and to cure disease, why would I study invertebrates like nematodes and flukes and the like? And the reason is because they are a major source of human illness. Now, this varies a little bit with regions of around the world, but certainly this is something that you would then have to understand. Now, nematodes are very important in disease, but also I'd just like to say that there's obviously a lot more to them than disease. They are one of the most abundant organisms on Earth. And once again, if you were to just go to a pond near you, take a drop of water and look under the microscope, you would see lots of nematodes. So if you look at a square meter of, say, fresh water, there could be 1 million nematodes there. And so they are often underestimated as far as the roles that they can play. And certainly they're very important in food chains. They eat lots of other organisms, and then a number of other organisms can eat them. Nematodes can vary a lot in size, and so therefore what they eat and what can eat them obviously would vary with that. There are lots of other types of invertebrates. So, for example, there's a group of shellfish known as brachiopods, and they have cousins bryozoans. And together, the brachiopods and bryozoans, they form a group called the Lophophorates because of this structure called the lophophore, which they would both share. So this structure used in feeding is a lophophore that both bryozoans and brachiopods have. If you look at fossils in the Paleozoic, these animals were extremely common. And so very often the bryozoans were colonial. So just like corals, you could have lots of little ones living together. They are not as common as they once were, but nevertheless, they can still be found. So for example, here is a bryozoan that one could find in fresh waters throughout the United States. It's a colonial animal, filter feeding from fresh water. The cousins, the brachiopods, once again, not as common today, but very common in the Paleozoic. So today we think of shellfish as being mollusks, things like clams and scallops and the like. But brachiopods are another type that make a calcium carbonate shell, but they're not mollusks. They're lophophorates instead. Okay. So as I go through diverse invertebrates, a number of topics come up, like segmentation did here. In the next video, I'm going to talk about the coelom and mollusks. So the coelom in general applies to many of these. So for example, annelids have a true coelom, nematodes have a pseudocoelom. So I'll talk about the coelom in the next video with mollusks, and then after that talk about arthropods.